For this video lecture, we're going to talk about a topic that's extremely important for those in healthcare and something we're going to be hearing more and more about. We're already hearing about a lot of this in the news, and we're going to continue hearing about it, and it's the problem of antimicrobial resistance. So we've talked a little bit about this already in the course, and it's ongoing problem that we need to understand more about why this is happening. All right, so here's a diagram from your textbook which illustrates how even in an artificial situation, like if you were working in the lab with some species of bacteria, you might be able to trigger resistance to a particular antibiotic drug in the laboratory artificially. And then while we're going over this, think about this happening inside human bodies all across the world as we take antibiotics for infections that we have. But what they're showing you, let's suppose you've got a population of microorganisms growing in the laboratory here on an agar plate. And what they're showing you here is the white colonies. Those are supposed to represent non-drug resistant organisms. But within that population, we see a couple of drug resistant mutants. And so back in chapter 8, we learned about how mutations can occur spontaneously, where you just have a improper nucleotide base incorporated into DNA during DNA replication. Then there are things that can induce mutations, but then don't forget those horizontal gene transfer methods that we learned about as well, like conjugation and transformation and transduction. So there are different ways that this diversity can occur, where you have a population of an organism where everybody's sensitive to a drug. The drug will kill you, but then within that population you will at times have resistant members that develop. And then what they're showing you here is, all right, let's suppose you add an antibiotic drug to petri dishes of this organism, and the drug winds up killing the population of microbes that are sensitive to the drug, not resistant to it. And what that does is it just leaves the resistant ones are the only remaining survivors. And then if you kept incubating that culture over time, these guys are going to divide and spread across the whole petri dish, and eventually you're going to have a plate full of cells that are resistant to that particular drug. So this is an artificial example of natural selection taking place. So imagine this occurring in human bodies all around the world. We have this arsenal of antibiotic drugs that we use for many different types of microbial infections. And whenever you take a drug, you're wiping out the members that are sensitive to the drug. But there may be resistant members inside your body when you take a particular drug. And if so, they're not going to die from it. Now hopefully, in a real world situation like inside your body, the idea when you're taking a drug like this is you're beating down the numbers, and then your immune system, which we will be studying as we get into chapters 12 and 13 and 14, is going to come in and hopefully finish off the rest. That doesn't always happen, but that's kind of the general idea. But meanwhile, you might be shedding those resistant cells out of your body and into the environment where they can be picked up by other hosts, and if they get transmitted to another person and that person gets prescribed that same drug that you were prescribed, it's not going to work for them because you have a resistant strain of the organism that has developed. All right, so again, this is an example of natural selection. It's also an example of evolution of organisms occurring on a smaller scale. So this is an increasing problem all the time. Back in the 1930s, 1940s, that's when we really started using antibiotic drugs to fight bacterial infections. And back then, people thought, this is great. We're developing this arsenal of all these different kinds of drugs, and we're going to wipe out infectious disease. This is going to become a thing of the past. But what people didn't appreciate, they didn't know about how genetic mutations work. They didn't know about horizontal gene transfer back then, and so nobody really expected how quickly microbes would be able to mutate and change to become resistant to these various drugs that we have. So it's an ongoing battle that takes place where We throw drugs at them, they mutate and change and become resistant, and then we got to throw other drugs at them. And that's something that's just going to continue and continue and continue for the foreseeable future. And I could go on and on all day about this. I'll try not to, but think about it as well. You may wonder, why don't we just develop new kinds of drugs? Well, that drug development takes a lot of money, and a lot of these antimicrobial drugs, not all of them, but many of them are fairly inexpensive. So it's difficult for pharmaceutical companies to get their costs back that are associated with researching for new drugs and developing those new drugs and showing that they're safe and going through all the testing. That's all a very, very expensive process. All right, so what are some mechanisms of antibiotic drug resistance in bacteria? Now here we're focusing in on bacteria, and some of these things apply to other microbes as well. But first of all, not all antibacterial drugs are going to work on every species. I think we all understand that at this point, because sometimes an organism, a particular species, just has a natural resistance to a drug. The drug can't get in the first place is usually what that is. So a lot of times it's based on structure. There are differences between gram-negative and gram-positive bacteria in terms of how sensitive they are to many drugs. In general, there are fewer drugs that work on gram-negative bacteria versus gram-positive, and it's because the gram-negative bacteria have that outer membrane. So they've got that extra layer, and if you guys remember back from chapter 3, that outer membrane of gram-negative bacteria has porins in it, which are protein-based pores that things have to pass through if they're going to be able to eventually make their way to the inside of a cell. And so if gram-negative bacteria don't have porins that will permit a particular drug to pass through, that drug's not going to have any effect on that particular organism. Now, initially, the naturally occurring antibiotic drugs that work on bacteria, many of those were not effective against gram-negative bacteria simply because they could not get inside the cell. But remember, we have semi-synthetic, so the chemists can tinker with those and change them, and that's one of the reasons why they do that. It's to modify them so now they can pass through a porin and go to the inside of the gram-negative bacteria. So some of that tinkering that goes on has made some of those naturally occurring drugs to be actually effective against gram-negative bacteria. Or you may lack the target of the drug, so a drug may act on some particular enzyme that doesn't exist in some species. Transporters. All right, so we just talked about the outer membrane, and a porin would basically be a type of transporter, but then think about your cell membrane, the innermost layer around the outside of the cell. Most antibiotic drugs are not going to, or antibacterial drugs are not going to freely pass across a cell membrane. They can't get through that phospholipid bilayer, so there's got to be some sort of a membrane protein through which they'll pass. That's going to be a transporter. Just kind of like a nutrient has to have a transporter protein to get in, like glucose or an amino acid. And then there are membrane proteins called efflux pumps that exist in the membranes of cells. That's what they're showing you over here on this diagram. That's supposed to be an efflux pump protein. And so what those pumps will do, let's say an antibiotic drug comes into the cell, these efflux pump proteins turn around and pump them right back out to the outside. And so those are naturally occurring membrane proteins that are likely involved in pumping waste molecules out, but because of genetic mutations can alter or change proteins, and so maybe now they'll also pump out an antibiotic drug they couldn't pump out before. Then acquired resistance, this is where DNA mutations occur, or drug resistance genes are picked up through horizontal gene transfer like we've talked about in the past. If you look at some of these other things on here, we say an antibiotic resistance gene. So remember, sometimes those are on plasmids. That's what we see here. Those are little extra pieces of circular DNA that some strains of a bacterial species will have. And so the gene on the DNA might code for an enzyme that degrades the antibiotic, breaks it down, or it might code for an enzyme that alters the antibiotic so that it doesn't work anymore. So there are different ways that that microorganisms can wind up destroying or altering antibiotics that we are trying to use to fight them with. All right, something I want to point out to you, because working clinically, you may hear this term used, especially around the clinical microbiology laboratory. You might hear about a patient having an infectious organism that is an ESBL. And what does that actually mean? So an ESBL stands for extended spectrum beta-lactamase. All right, so if you guys remember when we were going over a couple of video clips ago, we talked about beta-lactam antibiotics, penicillin and its relatives. Those are all cell wall synthesis inhibitors in bacteria. So these are antibacterial drugs. So beta-lactamases, there are different versions of these. These are going to be enzymes that destroy beta-lactam antibiotics. So it's got the ACE on the end, so it's an enzyme that destroys the antibiotic that we're trying to use to treat the infection. So some of these can be very restricted. They might destroy, let's say, penicillin and nothing else, and sometimes that would be called a penicillinase. But the beta-lactam antibiotic is pretty extensive. There are lots of different versions of those. Some of those are naturally occurring antibiotics. Some of them are semi-synthetics. We now have bacterial strains out there that are producing what are called ESBLs. These are extended spectrum beta-lactamase enzymes that destroy essentially any antibiotic drug in that category. And that's a big category of antibiotic drugs that has traditionally been used to fight bacterial infections. So if you become infected with a strain that produces this type of enzyme, it really cuts back on the arsenal of drugs that we have available to treat the infections. And so these are a growing problem across the world because we just don't have as many drugs to treat patients with when they wind up getting infected with one of these strains. All right, so there are genes encoding ESBL enzymes. So this is an enzyme, so it's going to be a protein. So there are bacterial strains that have acquired genes that code for these, and those have been identified in Klebsiella pneumoniae, which is actually, believe it or not, an intestinal organism that lives naturally in the intestinal tracts of lots of people. But as the name suggests, if it gets into your lungs, it can cause opportunistic pneumonia in that location, among some other things. You're more likely to get that, let's say, if you're hospitalized and let's say you're intubated. Klebsiella oxytoca is a close relative. And then good old E. coli, Escherichia coli, there are strains of this that have been identified as producing these types of enzymes. And so if good old E. coli gets into your urinary tract or down into your lungs if you're hospitalized and on a respirator, or it gets into your blood, there may be a very limited panel of antibiotic or antibacterial drugs that we can use to treat those infections. Also identified in other members of the Enterobacteria ACE family. That's a family of bacteria that includes these guys, as well as Salmonella and Proteus. These are all intestinal organisms that are gram-negative, but when they get out of the intestinal tract into other parts of the body, they can cause some pretty nasty infections. Also Pseudomonas aeruginosa, that's a gram-negative organism that's very common out in the environment. But again, it can cause infections in compromised individuals, especially people with weakened immunity. Causes a lot of infections in patients with burns and wounds of various types. So this is not good that we have this going on. And this is kind of a scary map. And this is actually from 2006, and it's showing the proportion of invasive Escherichia coli and Klebsiella pneumoniae resistant to third-generation cephalosporins. So a third-generation antibacterial drug means it's a more modern version. The cephalosporins are bacterial cell wall inhibitors. They're part of that beta-lactam group. And this was back in 2006. This was a growing problem at that time, and it's even worse now. But here's a map of Europe here and over here. And so when they were testing E. coli from patients that had E. coli infections, or Klebsiella pneumoniae from patients who had infections with that organism. In some parts of Europe, over here in the eastern part, over 30% of the Klebsiella pneumoniae were ESBL producing Klebsiella pneumoniae. And it looks like about 20 to 30% of the E. coli from patients that were in that category as well. Strains that were making these nasty enzymes that destroyed the members of that beta-lactam antibiotic family. So not a good thing. That's 2006, so that's actually 9 years ago. And so the problem is no doubt getting worse now. There are lots of examples of this. You'll hear about carbapenem. If you go back in your chapter 10, there's a table showing you different antibiotic drugs, and there's a group of related beta-lactam antibiotics called the carbapenems, which are one of the stronger, more powerful types of beta-lactam antibiotics, and we now have strains of various bacterial species that are resistant to carbapenems. So you're going to hear about carbapenem-resistant bacteria as well. And the situation just keeps getting worse and worse and worse. And this is one of those things that's really interesting to talk about. We could go on all day about this. And something to think about too is, where is this coming from? In some parts of the world, especially in poor parts of the world, there's no regulation on antibiotic drugs or semi-synthetics, any of these antibacterials. You can go walk into the grocery store or whatever and buy pretty much whatever you want. So there are people taking those drugs at times when they really don't need them. Even in the United States, sometimes antibacterial drugs get prescribed to patients who don't even have a bacterial infection. They might have a viral infection. Agriculturally, these drugs are used to try to improve the health of livestock, and then there's so much widespread use, those drugs wind up in our water supplies. And as long as they're around, they are causing natural selection to take place. You are selecting for the ones who are resistant to those particular drugs, and then they begin to dominate the population. So be sure you read your Inside the Clinic at the end of chapter 10. And, how do we combat drug resistance? Kind of like I was just mentioning, we shouldn't be prescribing antibacterial drugs to patients who don't have bacterial infections. Now, sometimes a physician doesn't know whether you have a bacterial or a viral infection, and just in case you do have a bacterial infection, they're going to go ahead and prescribe medication that will help fight that. And but that's a practice that the medical community is trying to get away from, because that's going to help us combat drug resistance. And as we get better at being able to diagnose and identify what exact pathogen is causing an infection, when we have more of these things like the rapid strep test or the rapid flu test and so forth, that's going to help us out as well. If a physician knows you've got influenza, which is a virus, they're not going to prescribe you an antibacterial drug, because that's going to do absolutely nothing for you. So as we get better diagnostics, that's going to help with this as well. Another thing too, when you are prescribed an antibiotic drug for an antibacterial infection, you want to be sure that you take all of your medication like they tell you. Because the tendency can be, sometimes you're given a prescription, say, for 10 days, and you might be feeling better after 5 days. Well, I don't need to take this anymore. I'm feeling better. But sometimes you have partially resistant bacteria that are part of the population infecting you. And if you kept taking the antibacterial drug for the prescribed amount of time, eventually that drug would wind up killing them and knocking them out. But when you don't, when you cut it off too early, you're giving those partially resistant bacteria, they survive, and you're giving them more opportunity to mutate and become fully resistant to the drug. So it is important that you finish a prescription. That does help us combat drug resistance as well. Another little side topic here, you've probably heard of probiotics. That's kind of a buzz word these days. You need to eat foods that are rich in probiotics. There are a couple of terms, probiotics and prebiotics. When you hear about probiotics, those are products like yogurt and other dairy products that have live microorganisms in them. Or sometimes you can even buy these as lyophilized or freeze-dried powdered bacteria. But that word probiotic is referring to live, usually bacteria, that are meant to make sure you're keeping, usually it's the intestinal tract, populated with happy, friendly microorganisms that should be there. So interestingly, quite a few of the types of bacteria that are used to make yogurt and other dairy products also naturally live in our intestinal tracts, and they're friendly microorganisms there. And that's important for microbial antagonism, where these nice friendly microbes are hostile to the growth of pathogens. You know, a lot of that's just about taking up space. You want these friendly bacteria taking up all the room down in your colon so that there's not any space for pathogens to come in and attach and start feeding off of the nutrient supply there and making you ill. So probiotics are live cultures. Prebiotics, those are not live microbes. Those are nutrients that are thought to encourage the growth of beneficial microbes. Lots of research being done in this area. There are some types of sugars that are apparently function as prebiotics and tend to be like a preferred food for friendly microorganisms, which we'll talk more about that as we get into chapter 11. There's some thought, there's a lot of research into diet. You know, how does your diet that you eat, how does that affect these friendly microorganisms that are living in your intestinal tract and other parts of the body? So there's a pretty good body of research out there now that when you eat a healthy diet with lots of fresh fruits and vegetables and so forth, that promotes the growth. Those are kind of prebiotic type foods that promote the growth of healthy organisms on and inside your body. When we eat a crappier diet that's not full of good, rich nutrients and is more junk food oriented, that seems to maybe promote the growth of microorganisms on and inside our body that are not so helpful. So that's a big hot area of research, and we'll be hearing a lot more of that as we continue on into the future. And also when we get into chapter 11, we're going to talk more about these normal microbes and where they exist on and inside your body. All right, so that video clip wound up being a little bit longer than I expected because I tend to get on my soapbox over that topic. I can't emphasize enough how important that topic is for people who are going into the medical area of any kind. All right, so the next video clip for chapter 10, we're going to talk about some interactions between drug and the host. So what happens, there are cases where you take a drug and you don't have a good reaction to it. So we'll talk a little bit about that and why that occurs. And then that should wind up chapter 10 for us.